Okay. Hello, everyone. Thanks for joining us. It's great to have you all with us this evening. Our theme this month, as you know from our social media and emails, is on nutrition and diet. And this evening, we will have the first live session of the month as a Q&A session. And our guest is all the way from Ireland. So we have Liam Walsh with us. Liam is a PhD student in Professor Paul Cotter's lab in Ireland at the Agricultural and Food Development Authority. And Liam's PhD project focuses on fermented foods and food production and processing facilities. So we're going to learn about this. We have prepared some questions for Liam, and we will be going through them in the first half an hour or so. And after this, we will have a 10-minute break, a comfort break. And during this time, you are more than welcome to submit your questions. And you can also submit your questions using our YouTube chat function on our channel. So, like I say, during the Q&A session, if there are any questions raised, please just drop them into the chat. And if you asked a question and Liam answered it and you still have got some questions or you want to ask a follow-up question, please just feel free to drop them there. So, Liam, thanks very much again for agreeing to be with us today. I'm very pleased to host you. So would you like to start by introducing yourself to us briefly?
Absolutely. And as you said, I'm a second-year PhD student and I'm studying fermented foods, namely kefir and the kefir microbiome. So we're identifying the microorganisms that are in kefir and we're identifying their functional aspects to see what's driving the fermentation process. And we're trying to link that to aspects of health.
Great. So obviously there is a lot of talks going on about microbiome, gut microbiome, human microbiome. Everybody describes it in a different way. So can you tell us what type of microbiome you're working on? Is this the microbiome that we're thinking about? And what it actually means? What is the definition of the type of microbiome that you're working with?
So I'll start with the definition of the microbiome. So you might be surprised to learn that we're actually not alone in our own bodies. So inside every person are trillions of microorganisms. So these are bacteria, viruses, and fungi, as well as other life forms that are collectively known as the microbiota, and their collective genomes are known as the microbiome. So there's a distinction there. Various environments, like for example, the soil, the ocean, and human bodily sites, have these distinct microbial inhabitants, but the one that's acquired the most attention in biomedical research is the one in the gut, which harbors the most of the microorganisms in our bodies.
Okay. So can you tell us why microbiome is so important for our health?
So we've evolved to live with these microorganisms for millions of years, and it's being increasingly thought of as a virtual organ. And if you just compare the genes in our microbiome to the genes in our own genome, they roughly outnumber the genes in our own genome by 150 to 1. And just to explain genes are informative sections of DNA, and they tell the cells to do things. So they can act as a blueprint and tell the cell to produce a particular protein. And so they're carrying out a lot of functions. The amount of genes as opposed to the amount of genes, our own genes as opposed to the human microbiome genes, are roughly 150 to 1 in favor of the microbiome. So they're carrying out a lot of functions. So it's not surprising that they have an impact in health. And these gut microorganisms have impacts in improving health outcomes. For example, they can stimulate the immune system, they can prevent the establishment of bad bacteria in your gastrointestinal tract, and they can feed the cells in your body. They provide a mechanistic understanding into this. Cells in our gastrointestinal tract will consume non-digestible dietary fibers. So these are fibers that we can't break down ourselves, but the microorganisms in our body can. And they use this as nutrients divide and grow, and in return they produce byproducts such as short-chain fatty acids and gases. Short-chain fatty acids have been implicated in health, and there's three main short-chain fatty acids produced. This is acetate, butyrate, and proteinate, all of which have anti-inflammatory properties. Glutamate can feed, for example, cells in your colon, it's called colonocytes, and they have a really important functionality in that they can kill cancer cells. And acetate, the most abundant of the short-chain fatty acids, is a central metabolite required for other bacteria to grow. And they can also reach peripheral tissues and play roles in, for example, cholesterol metabolism, as well as regulating central appetite. Finally, to conclude, there was a randomized controlled trial that correlated the increased production in short-chain fatty acids with a reduction in dietary induced obesity, as well as reduced insulin resistance. So while the analysis was quite weak and that was only a correlation study, there is definitely good trends being identified that are saying that these gut microorganisms have an impact on health.
Okay, that's great. So can you tell us, this is very interesting, you work on the microbiome, but how do you actually investigate the composition of a person's microbiome? What type of techniques do you use?
Well, the first thing we need to do is we need to collect a sample. So we'll collect, for example, vaginal, oral, or nasal swabs, as we can collect skin scrapes as well, if you want to sample those bodily sites. But typically for the gut microbiome, we'll collect either a stool or a kind of a poo sample. And then we'll extract its DNA. And just to explain DNA again, because I think it's important to the next stage of the methodology, DNA is a sequence of nucleotides, and you can think of these as letters. And the way these nucleotides are arranged determines the genetic information they encode. So for example, in a human, this might determine what color eyes or hair we have, while for a fermenting microorganism, it might determine its ability to make a particularly tangy sourdough or a particularly flavorsome cheese. DNA sequencing, we then apply a DNA sequencing methodology, which it's basically a machine that reads these genetic letters and it generates a massive volume of information, which is interpreted using computing methods to tell us things like what microorganisms are there and what they're doing.
Okay, so you talked about DNA sequencing. So can you tell us a bit more about that?
In terms of the methods, like we run a DNA sequencing
Yeah. So
center.
how does the method work?
Well, that's quite complicated, and there's a large number of machines that do such tools. For example, you can have long read sequencers and you can have short read sequencers, but typically they'll work by you'll shear your DNA, so your DNA is double stranded. We'll shear it to a single strand, and we'll attach complementary DNA to that, which is tagged with a fluorescent labeling. And by detecting the fluorescence that's emitted after it binds to that DNA, we can detect which nucleotides were present there. And again, those nucleotides will tell us the genetic information that those microorganisms encode.
Okay. So basically, you just find out the code of the DNA
Exactly.
of these microorganisms. You just tell, how these letters are ordered, and from that, based on the maps that we have already, you can decide what type of microorganisms are present. You can identify, what they are. So,
it is as simple as that. It's reading a series of 4 letters repetitively and a large scale, because there's massive numbers. I think in a standard like a bacterial genome, there's anything between 100 so with 1,000 and 3,000 base pairs, which is those nucleotides. So it's done on a large scale.
Yeah. Okay. So then, so you read them and you map them to already known, information, and you determine what they are and which one, what type of bacteria they are, so on and so forth. So does it mean that our genetics may also play a role in the types of microbes that live in your gut?
So the relationship between our own genes and the microorganisms that live in our guts is relatively unknown. Indirectly, our genes can determine our diet preferences and our taste perceptions, which may indirectly modify the microorganisms that live in our gut through diet. And there was a study done on identical twins. So they analyzed the microorganisms in these identical twins, one of whom was healthy and the other whom was obese. And they observed that they had completely different microorganisms in their gut, despite the fact that they're identical twins. So if there is an impact from genes, it's not huge. There was another study that identified genes that they believed may regulate the gut microbiome. So they observed that individuals with a specific variant of the gene LCT, now this is a gene that influences if a person can digest lactose, which is a sugar found in milk as well as other dairy products. And they observed that individuals with this variant had a higher proportion of bifidobacterium in their gut microbiome compared to controls, suggesting there may be an effect with in terms of our genes on which microorganisms persist in our gut microbiome and which are more dominant compared to others.
Okay, all right. That's great. Thank you for that. So in this case, then, what we're saying is that environmental factors, which is what we eat, is more likely to dictate the type of microbes that we have in our gut.
Absolutely. Diet is really important in shaping our gut microbial composition. In fact, even changing up your diet can result in a change in your gut microbial composition in only a matter of days. And this is because it's providing the microorganisms, our diet is providing our microorganisms with the nutrients they need in order to divide and grow. So a wholesome diet that's rich in a wide variety of nutrients will provide these microorganisms with a wide variety of nutrients, and you're more likely to have a diverse gut microbiome.
Okay, so then I'm going to ask a little bit of a challenging question then. So can you, and it's okay if you prefer not to discuss this. So can you give us a bit more insight about, so if a person has got intolerances, then
Like,
what
for
has
example,
gone wrong in their microbiome?
well, that is a very difficult question, and it does come down to the fact that no individual person has the same microbiome. So we haven't even identified what is a healthy microbiome and what is an unhealthy microbiome. So we have no set resolution of, oh, there's set amount of species needed in the microbiome to confer overall health. So, for example, an individual with one microbiome that's healthy at the time may be not healthy for another individual. In terms of, let's say, excess antibiotic usage, resulting in a C. difficile infection, there is some methods where they transfer the microbiome called a fecal microbial transplant, but it's quite a blunt method where they essentially transfer a poo sample into an individual, and that can result in fixing the C. difficile infection. But I couldn't tell you if, as a result of, for example, excess consumption of processed foods, that you have certain bacteria that are causing your disorder.
Okay. Thank
We
you
don't
for.
have that resolution. I'm sorry.
Yeah, thank you. Thank you for rising to the challenge. So can you mentioned about healthy eating and its effect on the microbiome. So obviously fermented foods are the new trend. Everybody is, drinking either kefir or kombucha and things like that. So how can you tell us a bit more about how, do fermented foods help the microbiome? Why all of a sudden they're very popular? And whether or not we should actually use them in our diet to help the microbiome.
Well, firstly, they have a prebiotic effect. So some things like fermented foods, particularly fermented plant foods like kimchi, will contain fiber, which will fuel the microorganisms in your gut. And there's been a number of intervention studies, I think 19 to date, on fermented food consumption and modifications of the gut microbiome. But currently, the data is too sparse to suggest that the microbiome modifies, or that consumption of fermented foods modifies the gut microbiome according to a specific manner. There was a study that analyzed intake of fermented vegetables on a weekly basis to individuals who didn't consume fermented, or they didn't adhere to, or they didn't adopt fermented foods in their diet. And they noticed that their gut microbial profile was similarly diverse. So if there is impact on consuming fermented foods on your gut microbiome, it's not major, it's not profound. But there are subtle associations. So, for example, that study identified subtle associations between specific microorganisms and fermented food consumption. So there is this maybe a subtle change, suggesting there may be a subtle change. When it comes to fermented foods, it really boils down to it enhances the nutritional profile of the food. So if you think of, let's say, soybeans, for example, soybeans are rich they have a lot of indigestible proteins that we can't break down ourselves, but these fermenting microorganisms can. And by the process of fermentation, these microorganisms break down these complex proteins into simpler amino acids, which we can subsequently consume. So fermented foods are rich in a wide variety of nutrients as well as vitamins and minerals, which can modify our gut microbiome through diet. If I know I'm going on a little bit, but there has been very little health claims approved in fermented foods. So there's a large amount of studies out there that are claiming positive effects like anti-carcinogenic properties, but they haven't been verified with vigorous scientific testing in the form of large-scale human-specific clinical trials. And unless they're backed by such vigorous science, they can't be verified.
Okay, thank you for that. So, if that's the case, then so I know that you mentioned your PhDs on fermented foods and their effect on microbiome. So, can you tell us a bit more about your key findings so far, and what is the direction that you're going to take from this point onwards?
Absolutely. So we applied the same approach that I just discussed for analyzing a person's microbiome to kefir, but we did this on a large scale. So we analyzed 256 kefir-based samples, and we identified what microorganisms were there. And we determined that it was roughly, we determined that it was primarily a bacterial-driven fermentation, with a higher proportion of bacteria, typically accounting for 90% onwards of the total abundance of the sample, the total amount of microorganisms in the sample. And it was a smaller subshed section which were contributed to by yeast. And we identified keystone species that were driving the fermentation process. So typically, for example, Lactococcus lactis accounts for 80 to 90% of the total community present in the kefir fermentation, and it's carrying out a lot of the functions that are driving the fermentation process.
Okay. Okay, so you said you have looked at you sequenced the genome of the microbiome. So have you actually taken it from one person, several people, or where did you get your samples from?
For the kefir, okay, so firstly, kefir is a fermented dairy beverage. It's been described as a light yogurt, and it's produced from these things called kefir grains. So these are protein matrices that house the fermenting microorganisms. And some say they resemble cauliflower. And you simply place them into milk, and you wait for a period of 24 to 72 hours. After that time has elapsed, your fermentation is complete, and you subsequently have a fermented product. So what I did was I asked individuals in the fermenting food community, so enthusiasts, would they kindly donate their grains to my study? And they sent them to Ireland using standard postal services. And then I grew up those grains till they were sufficient mass to grow in 200 milliliters of milk. And then I carried out the fermentations in the lab to collect samples for sequencing.
Okay, okay. And when you say sequence, again, can you tell us what type of sequence was that? Could you actually do it with a simple sequencing reaction, which is the one that we described? Or did you needed something a bit more global, a bit more sophisticated to process these samples?
So we used short read sequencing. So we read small individual nucleotides. And from those small individual nucleotides, we try and make, we have a sequencing center here at Chagas. And basically we prepare samples. So we fragment up the DNA, and then we individually sequence those small little fragments. And then in later stages, we try and piece these little fragments together similar to a jigsaw puzzle. And from that, we can tell things like what genes are there. And if we're good enough, we can assemble the entire genome of the microorganism.
Okay, okay, that's interesting. So you mentioned that there has been some studies which looked at the effect of fermented foods on the microbiome and whether or not they could be used for easing any conditions, any health conditions, health problems. So, are there any health problems that you think fermented foods can help with? So, for example, do you think intolerances might be improved by using fermented foods?
That's a great question. And there's only one verified health claim approved under EU regulations, and that relates to live yogurt cultures on improving the symptoms of improving lactose digestion. So that's one that's been verified. Certainly, there's good evidence to suggest that they have other roles as well in certain aspects of health. But it's certainly in my own research, the more I study fermented foods, especially fermented food microbiomes, I realize that they're individual cases. So every microbiome is a singular entity. So even if, let's say, a particular kefir grain or a particular kefir is associated with, say, reduction in cholesterol, If you're using another kefir grain, they might have different microbial species that don't have the same functionality, so they won't have that cholesterol reduction characteristic. So there's a lot of variability, particularly with household-based fermentations, which is why industry is trying to go into isolating individual strains to produce a more uniform product.
Okay. So the other thing that I'm wondering is, obviously, long-term antibiotic use is one of the main reasons that actually sort of destroys the balance of the microbiome in the gut. Is there any disease or a condition or a drug use you think will damage the microbiome forever, or no matter what the damage is, if you treat your microbiome well, or if you eat the right products, right food, fermented food, or whatever, can you actually regain that healthy microbiome back?
So it does boil down to diet. The more you eat, the more likely you're going to have a diverse and good microbiome. Antibiotics can certainly have a negative effect. So they're drugs that they're used to treat illnesses and diseases, for example, and they work by either killing these microorganisms or preventing them from multiplying. And over the past 80 years, they've saved millions of lives, but they do kill indiscriminately. So they do kill good as well as bad bacteria. So they do have a harmful effect on our microbiome. Would you repeat the question? I think I forgot it.
Okay, all right, lovely. Well, thanks very much, Liam. I made you to talk for about 20-25 minutes there. I think it's time to give a break and let you to have a rest. And then during the break, I will be collating the questions that our audience have submitted so far. And I will be asking these questions to you on their behalf when we resume back. So shall we come back at 7:05 and then carry on with this lovely discussion? Thank you. Hello, everyone. Thanks for keeping or staying with us during the break. And we're now back to answer your questions. So, Liam, I have got all my questions on my phone over here. So I'm going to start with the first question that we got. So the audience is asking, have there been any studies that have demonstrated a link between COVID and the microbiome?
Yeah, there has actually, in fact. And so there has been implications in, for example, having a less diverse gut microbiome and prolonged symptoms of inflammation that are associated with long COVID. So what it is the gut microbiome is one of the main immunological organs in your body. So it does have an effect in moderating or controlling the magnitude of your immune response towards COVID-19.
Okay, that's very interesting. So is it a one-centered study? Is it a multi-centered study, the one that you're mentioning?
It is a full paper, and it does look at the gut microbial diversity between individuals. So it looks at the gut microbiome between individuals who have received COVID, and it looks at them compared to, so if they have a milder symptom of COVID as opposed to a more severe with long COVID, and healthy controls. So
Okay.
it's a full-blown paper.
All right. Okay. So is this a UK only study, or is this an international effort?
I actually can't remember the fine details of it, actually, but I can absolutely get it on in the. I can get it. I can get
Yeah,
the
that
study
would
to you in
actually be
the paper
quite
and everything.
interesting. We can always share it on the website on your page. So I'm sure people will be interested to learn more about that. So another question from the audience again. So because there is so many types of microbes in samples, is it hard to map the DNA back to a reference?
That is a brilliant question, and it absolutely is. If you're collecting, for example, bodily sites, they're often contaminated with host DNA, and it can be really hard to get rid of all the contaminating host DNA and collect microbial DNA or microbial reads. We're lucky with a poo or a fecal sample because it has a high bacterial load, so it's a little bit easier to collect microbial samples from there or microbial DNA from that.
Okay, all right. That's interesting to know. Thank you for that. So does the extent of fermentation affect the impact on the microbiome?
Like for how long? I suppose they mean how long it
Yeah.
is it? So on average, you will be. I suppose that comes down to the exposure to microorganisms. So you're going from, let's say, for fermented milk, you're going from a milk profile which is relatively absent. There's a small number of microorganisms, and then there's a pattern. So I'll use kefir here because it's something I'm used to. There's a succession pattern, so a change in the microorganisms. So yeah, at an early stage of fermentation, the kefir process is typically dominated by, for example, Lactococcus kephinofaciens, while at 24 hours is dominated by Leucanostocks mesenteroides. And if you transition on into the 48 as well as the 64-hour category, you're running out of nutrients. So it selects for particular species over time. So you do get a selection. So you're exposing yourself to less microorganisms if you ferment for longer. And there's obviously there's a chance for a unwanted secondary ferment where it can disrupt the population dynamics. So it can expose it to, for example, spoilage-causing microorganisms.
Okay, so what you're saying is that actually fermenting it longer is not a good thing.
It can be in certain instances, but there is recommended protocols. For example, fermenting vegetables for 2 to 4 weeks. Doing kefir for roughly 24 hours or anything between 3 days. But if you go beyond the recommended fermentation time that's recommended by standard trial and error, got these time points, and if you go beyond them, you do run the risk of a unwanted secondary ferment.
Okay, all right, that's interesting to know. Okay, so the next question from the audience is, I can understand that fermentation can bring about beneficial changes in a dough, but there are also claims that the organisms help after bread is eaten. Surely they have been they have been killed by cooking? That's a very interesting question, I think.
They're related to sourdough, is it?
Yeah, I think so. I think so.
Absolutely. But if you think about it, these microorganisms, they're not likely to become established in your gut microbiome. So he is correct, especially in bread making, the heat will probably kill most of the microorganisms, but they will still produce those byproducts, which some may survive the heat treatment. And these microorganisms don't tend to become established in your gut microbiome. They don't easily become established. So your gut microbiome typically holds, I think, anything between 500 and 10,000 species. And it's really hard for a single individual species in not an unbelievable amount to become established in such a stable community. So I wouldn't be too caught up on if these microorganisms can become residents. I'd more focus on the nutritional profile of fermented foods in promoting health.
Okay, lovely, great. So, okay then. So there are a few other questions as well. If you're not too tired, I would like to keep going.
Absolutely. And I might just go on to that point because it was a really interesting one, just because I thought of something. There was another study as well that looked at the microorganisms that could be introduced from fermented foods into your gut microbiome. And there were, in cases like, for example, Streptococcus thermophilus, as well as Lactococcus lactis. So these are typical lactic acid bacteria that drive the fermentation process. And they were present in the gut microbiome. Now, whether they were long-term residents or they were just simply passing through is another story. But there is certainly evidence that they can colonize the gastrointestinal tract, or they could be short-term colonizers where they produce certain byproducts, but that hasn't been verified.
Yeah, I think what's very interesting from that question as well is the fact that, bacteria, they do have an optimum growth temperature, which is generally about 37. Obviously, you have got bacteria that grow on either extreme of that 37 degrees. But when you think about it, when you put the bread to 200 degrees Celsius oven, you do expect them to die. How do they stay viable though?
Are you meaning during the baking?
Yeah, in the bread, because like the audience is saying, in some instances they say that when you eat the sourdough bread, then you can get benefits for your microbiome. But if the bacteria is cooked, are they going to be alive to actually help your microbiome?
Yeah, and if you think of sourdough, you're using these bacteria as a riding agent. So using, for example, Saccharomyces cerevisiae or and as well as Lactobacillus sanfranciscus. And I'm not aware of the mechanisms with which they survive the baking process, but I do know that when you're consuming sourdough, you're getting a sour taste and you're also getting the bread to rise. So there is clearly activity there, and there's clearly microbial activity being driven on during the cooking process. So but that's more logical than a detailed understanding.
Yeah, lovely. Thank you. So one of the other things that I was wondering, I want to follow up on the last question that I asked before the break. So we said that, antibiotics can have an effect on the microbiome, and when I asked, can you actually reverse this process, you mentioned that, if you feed yourself with good food, you're more likely to get it back. So in addition to antibiotics, though, can you think of any other physiological events that might affect your microbiome or its activity? For example, as you age, does your microbiome age as well? Do you get any differences in your microbiome as you get older?
Absolutely, you do. You do get a loss of diversity as you age, particularly in older ages. You do get a loss of diversity. So initially, when you're born, your gut microbiome undergoes great microbial change, and typically around adolescence, early adulthood, your gut microbiome remains relatively stable, and it stays relatively stable from that age group to an adult. But there is decreases in the microorganisms in your gut over time. I don't know why that is. I'm not aware of it. Um, I know there was a study that analysed individuals in care homes, elderly individuals in care homes, and elderly individuals who were by themselves, and the individuals in the care homes had a lower gut microbial diversity, but they linked that to diet as well. Um, I'm not exactly sure why there is that decrease in diversity. It's definitely something I should figure out.
Okay, that's interesting. So in addition to that, though, obviously considering or relative to 3 or 4 decades ago, our lifestyle, even in our kitchens, is changed dramatically. I mean, we have got more processed foods, canned food, frozen food, so on and so forth. So do you think we are capturing sort of a evolution of our microbiome as our life goes by, or do you think it's actually getting worse? How do you consider all of this change in diet and any potential increase in microbiome related diseases?
Absolutely, yeah, you're dead on there. So we unfortunately we have lost a lot of diversity in our Western diet. So I think it's 75% of the world's food supply comes from 12 plants and 5 animal species. So there's considerable loss in diversity there. And so we're not getting the nutrients that are needed to feed the microorganisms in your gut. And they observed a study where they analyzed the gut microbial diversity in individuals living in rural communities, so rural Africa as well as South America. Um, and they compared those to those on a westernized diet in Europe as well, or those of the West on, for example, Europe as well as America. And they noted that they had reduced diversity in their gut microbiome. So the gut microbiome diversity was a lot higher in individuals from rural communities. And they linked that to diet because they had a kind of hunter-gatherer lifestyle where they consumed a high amount of fiber as well as plant-derived protein sources. So certainly diet is heavily an influence, and it is influencing our gut microbiome. In addition to this, we're applying more safety, we're living in a more sterilized environment, and we don't get as much exposure to microorganisms. So there's links between, for example, allergies. So they say that they identified a correlation in allergies between individuals with asthma, and they said that there was a positive correlation having a dog in your home, for example, because you get exposed to the fur and whatever, so you're less likely to develop extreme allergies as well. So it comes from having a hyper clean environment, not getting enough foods. We can also use antibiotics to treat a lot of infections, and it wasn't as safely managed as it is now. So there's a lot of factors contributing to that, absolutely.
Okay. And do you think public is more informed or more aware of what they're doing to their microbiome, or do you think there is a lot that needs to be done?
I think that there is good awareness, to be honest. Well, I come from a science background, but I didn't, prior to my PhD, even know what fermented foods were. But I was aware that there was microorganisms in our guts. And I think that the good thing about the microbiome is it comes down to healthy lifestyle. So as long as you apply a healthy lifestyle, you'll have good microbial diversity. And we already knew this anyway. So while it would be nice to tell more people that, look, you're not just a singular entity, you're more like a microbial village. It's not necessarily information they need, because they already know that, to promote health, you need to live a healthy lifestyle, or you need to take steps for a healthy lifestyle.
Okay, that's great.
That's my 2 cents on it anyway.
Yeah. But, can you tell me a bit more about, for example, if you have got intolerances which relatively, restricts your diet significantly, what would be your advice for those people to keep their microbiome healthy?
As in terms of well, see, this is the great thing, is that there's so many foods out there that are high in fiber. So all you need to do is you need to consume some kind of dietary fiber, and you need to plant-derived protein sources, for example, as well. So like, for example, vegetarian diets, they tend to have higher gut microbial diversity because they have more plant-derived protein sources. So as long as you're eating something healthy, and there's a wide variety of nutrients that contain fiber, like fruits and vegetables, as whole grains. So I'd be very surprised if there isn't foods out there that contain the necessary amount of fiber, regardless of dietary restrictions. I know, for example, that fermented foods can be high in histamine, for example. And so people that have histamine intolerances have come to me and they've said, should we be consuming fermented foods? And I can only really say that you can't really consume fermented foods. Like, some studies have shown that, for example, kimchi can have effects on reducing the levels of histamine, but they've not been verified. So I think that's one particular issue where I've kind of gone, well, you can't incorporate fermented foods into your diet. But that's the only one I can think of.
Yeah. I mean, I guess it's easier when you have got, one type of intolerance, but when you are lactose intolerant, gluten intolerant, all in together, it just makes your life a lot harder, I suppose, in terms of fiber intake as well.
Yeah, absolutely.
Okay.
I'm sure there's alternative solutions. Unfortunately, I wouldn't be I pretty much eat everything, so I wouldn't be something I'm too conscious of.
Lovely. Well, thanks very much. This was a lovely discussion, and I'm sure we all learned something from this discussion. It was very interesting. Thanks very much for your time again.
No problem. Great talking
And
to you.
everyone, see you next time. So our next session will be on Tuesday, next week Tuesday, and we will have a live talk on, again, a topic related to diet and nutrition. So see you all again.